Hello friends, today we will study about flagella. This is the third video of microscopy and morphology of bacteria. So let's begin from flagella. Flagella are cytoplasmic appendages protruding through cell wall. These are thread-like structures composed of a protein that is flagellin 5 to 20 micrometer in length and 0.01 to 0.02 micrometer in diameter. Again telling 0.01 micrometer to 0.02 micrometer in diameter. They are organ of locomotion. All motile bacteria except spirochetes possess one or more flagella. Now let's know about the parts and composition. Each flagellum consists of three parts: filament, hook, and basal body. The filament lies external to the cell and connected to the hook at the cell surfaces. The hook basal body portion is embedded in the cell envelope. The basal body contains outer and inner rings by which the basal body is attached to the cytoplasmic membrane. Outer rings are absent in gram-positive bacteria. The flagella is made up of protein, that is flagellin, which is similar to myosin. Although chemical composition of different genera of bacteria is similar, they are antigenically different. Specific flagellar antibodies are produced in high titrates in response to antigenic stimulation of flagella. These antibodies are useful in serodiagnosis but are not protective. Now let's know about arrangements or types. First is monotrichous, single polar flagella, that is at one end. Example, Vibrio cholerae. Second is amphitrichous, single flagellum at both the ends. Example, Alcaligens fecalis. Third is Lophotrichous, that is topped or topped of flagella at one or both ends. Example, Spirilla. Peritrichous. Peritrichous is flagella. In peritrichous, what happens? Flagella arranged all around the cell. Example, Salmonella typhi. Now let's know about the demonstration. Flagella are about 0.02 micrometer in thickness and hence beyond the resolution limit of the light microscope. There are some demonstration. Let's know about one by one points. First is dark ground illumination. Second is special staining techniques in which thickness of flagella is increased by moderating. Third is electron microscopy. Fourth is indirect methods by which motility of bacteria can be seen or demonstrated. Here there is 2 points. First is spreading type of growth on a medium. Example, swarming growth of Proteus species. B. Next, second point is motility under the microscope. Example, hanging drop preparation. Now, third point is spreading of a bacteria in semi-solid agar. Now, let's know about the types. There are mainly three types of fimbriae. Common pili. First is common pili. These are of 6 types depending on their morphology, number per cell, adhesive properties and antigenic nature. Sex are F. pili, that is fertility pili, col 1 collision pili. Now let's know about the functions. Adhesion. Fimbriae are organs of adhesion. This property enhances the virulence of bacteria. Transfer of genetic material. Sex pili are present in male bacteria. These pili are longer 18 to 20 micrometer and 1 to 4 in number. They help the male cells to attach with non-male or female cells, informing conjugation tubes through which genetic material is believed to be transferred from the donor male or recipient cells. Now let's know about detection of embryo, electron microscopy. Second is hemagglutination. Many fimbriated bacteria, example Escherichia coli, Klebsiella, strongly agglutinate red blood cells of guinea pigs, fowl, fowl horse, fowl horses and pigs, human and sheep cells weakly, and ox cells scarcely. This property of hemagglutination is a simple method for detecting the presence of embryo. The hemagglutination can be specifically inhibited by D-mannose. Now let's know about bacterial spore. Spores are highly resting stage from the formed in unfavorable environmental conditions, presumed to be related to the depletion of exogenous nutrients. As bacterial spores are formed within the parent cells, these are called endospores. Sporulation is not a method of reproduction. In the process of sporulation, each vegetative cell forms only one spore, and during subsequent germination, each spore gives rise to only one vegetative bacterium. Now let's know about morphology of spores. The clear area in the protoplasm of cell becomes gradually more opaque with condensation of nuclear chromatin forming the spore. The cell membrane grows inward and forms spore wall around the core. The innermost layer of the spore wall forms the spore membrane from which the cell wall of future vegetative bacterium develops. Outside this membrane is thick layer, the cortex, and the multilayered top spore coat. Some spores have an additional, apparently rather loose or undercovering called exosporium. Now let's know about the shapes and position of spores. The precise position, shape, and relative size of spore are constant within a particular species. Spores may be central, subterminal, or terminal. They may be oval or spherical in shape. The diameter of spore may be same or less than the width of bacteria, example bacillus, or may be wider than the bacillary body, producing a distension or bulge in the clostridium. Resistant. Bacterial spores are extremely resistant to ordinary boiling, disinfectants, and heating. However, spores of all medically important bacteria are destroyed by autoclaving at 121 degrees centigrade for 15 minutes. Methods of disinfection and sterilization should ensure destruction of spores also. Now let's know about germination. The process of conversion of a spore into vegetative cell under suitable conditions is known as germination. The spore losing its refractivity and swells when transferred to conditions conducive to vegetative growth. The spore wall is said to the germ cell appears by rupturing the spore coat. The formation of vegetative bacterium occurs by elongation of germ cells. Now let's know about demonstration. Germ staining. Spores appear as an understanding refractile body within the cell. Modified Zell nil insert Zn staining. Spores appear as acid fast, that is red in color. Zell-Nelson staining with 0.25 to 0.5% sulfuric acid instead of 20% sulfuric acid as used in conventional methods. as decoloring agent is used for spore staining. Now let's know about the use of spores. Spores of certain species of bacteria are employed as indicator for proper sterilization. Example, Bacillus Stereothermophilus, which is destroyed at a temperature of 121 degrees centigrade for 10 to 20 minutes. Same temperature and time as used in autoclaving. These spores may be kept in autoclave prior to its use. Absence of the spores after autoclaving indicates proper sterilization. Spore forming bacteria. Obligate aerobes genus bacteria Bacillus, example B. anthracis and B. subtilis. Obligate anaerobes genus Clostridia, example Cl. titani, Cl. welchi, Cl. Clostridium botulinum. Cl means Clostridium botulinum, like this, with bacillus and clostridium are gram positive bacilli. Finally, finished microscopy and morphology of bacteria. Any questions regarding this, you can ask me in the comment box, and subscribe my channel for more updates. Thank you.